Hello class, today's project is on skeletal muscle contraction. Our first step is going to be excitation. So the first step of excitation, our electrical stimulus is going to arrive at the axon terminal, and this is going to stimulate the synaptic vesicles to release the acetylcholine to the synaptic cleft, which is going to be our transmitter. The ACH is going to be our transmitter. So once it spreads across the synaptic cleft, it's going to bind to the passageways along the motor end plate, and it's going to allow for the sodium ions to attach and pass through the ligand-gated cation channels. So with the positively charged sodium ions passing through the canals, it's going to create a depolarization, which is also called the end plate potential. This brings us into our next step, which is going to be the excitation contraction coupling. The first step of this, we are going to be traveling along our sarcolemma, and the change in voltage created by the end plate potential from the previous step is going to begin opening the voltage gated sodium ion channels. So it's passing along here, and I couldn't draw them all open, but we're going to pretend like this keeps passing through and these channels keep opening, because the depolarization of one is going to initiate the opening of the next channel, and this is going to allow the sodium ions to pass through into the muscle fibers. This action potential is also going to reverse the polarity so that the inside is going to be positive in comparison to the outside. It then is going to pass down through our T tubules and to our terminal systema. They are linked to the voltage-gated protein, and once the T-tubes depolarize, these protein calcium ion channels channel in the terminal systema, and as a result, the protein opened the gates, releasing the calcium into the cytosol. So as the calcium ions begin to flood the cytosol, repolarization occurs in the sarcolemma, and the skeletal muscle contraction requires this process happening repeatedly, meaning that new action potentials are recurring, and with each one, the amount of calcium in the cytosol is going to increase, and that's going to manipulate us into our next step, which is our preparation for contraction. So when the calcium ions become available, they bind to the troponin and cause a change in the position. So this is actually going to move, and it's going to expose the active site on our actin. And so action is now going to be available to bind to the myosin head, and the muscle can contract via cross-bridge cycle, And this is going to lead us into our fourth step, which is the actual contraction. And so the myosin head is going to switch the previously attached ATP into ADP plus a phosphate. So the energy released from that is going to cock the myosin head to be erect. And this actually all happens before the active sites even become exposed. So once uncovered, the myosin heads are going to be ready to bind to the active sites. And this is the actual process that I was talking about, which is the cross bridge. So then as the ADP and phosphate detach, the energy from that, because once they bind to the site, it's going to kind of pull along with it. So once the ADP and phosphate is detached from that, the energy is going to be released. And once it's released, the head pulls. This is known as the power stroke. So the ATP molecule attaches to the myotin head, and the myotin head releases from the active site. So relaxation is going to occur once the contraction ends, and the calcium ion concentration is going to return to a resting level. And so once, I didn't actually draw a step for this because it has to do with the first step, I guess. So once the calcium ion level has rested, it's going to prompt for the ACH to quit being released, and the remaining ACH is going to actually disintegrate. The cation receptors are going to close, and the motoring plate is going to become in a resting state. The depolarization is then going to travel over to the sarcolemma, and the sodium gated ions are going to remain closed, and then the voltage gated potassium ions are going to open, releasing the potassium ions from the muscle fiber. This then continues down to the T-tubule, and the ions from the sarcoplasmic reticulum are then going to pass back through the voltage-gated protein. And once all these things have returned back to normal, that is the end of the relaxation period. Thank you.